Yeah, so thanks, Eric, for sparing me some time today to talk about
Always a pleasure.
your
Always a pleasure.
your career journey. Um, Eric, you're a Professor of Cancer Pharmacology and Molecular Imaging here at Imperial. And you're like me, a long time stalwart of Imperial. You started in 1998 and have been a professor
Yeah.
since 2006. So, like me, a 20-year veteran of the institution. But I'd really like to find out a bit more about how you got started in science. So, you were born in Ghana
Yes.
and did your first degree there. So you know, what was it like in your early years in Ghana? What were your ambitions growing up? Did you always want to be a
Yeah.
scientist, or how did you get into science?
Well, of course, as you start your undergrad and various, what you are looking for is some kind of career, and that was what was drummed into me. Find a career. And initially, I knew I wanted to do something in pharmacy or medicine, and and that was the starting point. But of course, I started the pharmacy degree and realized that I had a strong interest in research, in the sort of knowledge discovery elements side of things. So when I finished, I began looking for opportunities to evolve further. And some of the early things, and again, I like talking to people and trying to see where they sit. I don't always take the advice, but I listen to the advice, and sometimes I use some of that. And I think one of the pieces of advice I got was, it's important to, if you want to do more research, to then begin to understand technologies. And that really helped me because I did a master's in a sort of technology environment in pharmaceutics to try and gain more understanding then about spectroscopy and various. So those at the time, things like you call HPLC and various were very new technologies then. So trying to understand all these in some detail before starting a career in research was important to me, and I did that. And so I recall when I started doing my PhD, a number of things were very straightforward for me. I would walk into the lab and develop my method without going to bother my supervisor. And I think those are some of the elements that helped me right from the outset, delving into technologies, trying to understand the technologies prior to using them really helped me. So it was somewhat a sort of not as straightforward. I didn't get up and say, well, I was going to do a PhD. It evolved over time. Yeah, there was a time I was just going to go back and just work.
Right.
Did
you get a lot of Did you get a lot of support from family, from your tutors to go down this academic route and do further study?
I think probably more from just seeing how things went. Family were, do what you think is is possible. We'll support you regardless. So that was helpful. But I think in my, there were times when I had to make the decision, do I go back and just work and make money, or do I, remain in research and tag along with that and create new discoveries? And of course, I chose the latter, and haven't regretted it ever since.
Of all, now you've moved around quite a lot in your career, but of all the places you chose to take the next step from Ghana was Glasgow. So how did you end up?
Glasgow, yes. So, I mean, when I was looking at the time, I remember I had the options to probably go to Canada and start a research degree, or come to London at the time. But Glasgow had something to offer. So Glasgow had a very good technology program that I thought would help me in sort of pharmaceutical technologies. And so I moved there primarily because of that, because I felt if I could get a strong handle on technologies, then that will help me. And I think that has shaped the way I think about science. You know, really that a lot of science is led by developing strong technologies, which then move you. And we've seen that, you know, with things like CRISPR and various. You have very strong technologies, then you advance science in leaps. So
what's your abiding memory of your time as a PhD in the Beatson?
It was
stands
quite
out?
fun. You
It
know,
was fun.
I don't know if you know Glasgow and students in Glasgow, but it was fun. I enjoyed my time in Glasgow. We worked very hard at the Beatson. I think scientifically, I got there and some of the concepts were quite challenging. More the sort of biology side of things. And so I'll come in and from the sort of pharmaceutical side of things, straightforward, easy. And I had to really learn very hard most of the molecular biology that were ongoing at the Beatson. And, you know, the Beatson was a tough place at the time, you know, John Wyke and various and Workman and you know, you were given a hard time as a student to excel. You know, you did not have the option not to. You had to excel. If you did a presentation, it had to be A star. You did, you know, and that was it. It was a test of you and your supervisor. And A, you wanted to do well, and B, you didn't want to disappoint your supervisor. So it was fun. It was fun. We worked hard. We played hard. And it was quite enjoyable at the Beatson. I have very fond memories.
It's interesting how you describe the culture there. So you know the expectation that was put upon everybody to achieve. Do you think they got the balance right at that time? And do you think that that's the model?
I think they did. I think they did. The model was effectively these students, all students coming through the Beatson were going to be future leaders. And that was the sort of concept at the Beatson when I joined, you know. And so you were expected to learn not just the basic scientific, you know, stuff that you were doing, but also to learn things around it. So you were supposed to understand the cost of a mouse if you were doing mouse experiments, so that you were reasonable in your thinking as a future PI. And I think that balance they got right. And of course, a lot of the people who left the Beatson at the time all became PIs, you know. So I think they got it right.
So after your PhD, your next steps were across the pond to Johns Hopkins, to Baltimore, right?
To Baltimore, yes.
Which must have been an interesting city to be in at that time in history. What was the biggest difference between life in the UK and life in Baltimore?
Oh, a lot. So it was funny because the first day I walked into the lab, somebody ran in and said, you know, so that was a defining moment. You know, do I really want to be here?
Okay.
But I tell you what, Johns Hopkins is another really interesting place to learn. You know, there was no doubt that, you know, some of the top scientists, the most cited author in the world, the third most cited author in the world, were all at Hopkins at the time. And again, you were challenged. You went to grand rounds to present your idea, and you were shot at. And you know, you grew up thinking, okay, you know, either this idea is very bad, or I've got to really work hard and I've got to really work hard on it, or I need to really, revamp the whole thing. And when I sat sometimes, when I stood in front of a ground round to sort of present an idea, and I knew across the front row were about say 10 or so editors of journals, and they were all tough. And not unnecessarily tough. They picked holes in your idea and they told you so in no uncertain terms. This is lousy. I don't think this will work because of X, Y, and Z. And it was your position to actually defend that. And I think that's something we've lost in academia a little bit. People take things too personally when they are criticized and don't, mature sufficiently from some of the criticisms that are levied scientifically, that is. And I think we need to sort of get back to that route, because, for me, that was one of the brilliant things I learned from Hopkins. You were criticized, but also you were given a lot of good substrate for development. So we will have lectures as postdocs. We were lucky to be treated as students sometimes, and just like in Imperial and stuff on other times, but largely as students. So we will be able to attend lectures on best research methods, statistics underpinning research, how to write grants, being successful, an NIH grant, for instance, that sort of thing. Some of the early grants I got when I moved to Imperial were the US Department of Defense grants and NIH grants. And that was partly, the sort of learning of how best to develop your grant ideas. For instance, making sure that it's a 5-10 year idea and not just a sort of a 2-year idea. And those things, I think, helped me significantly when I became a PI.
So were those interrogations the toughest part of that early part of your career, or did you quite enjoy the combat?
At the time, as a young postdoc, it was tough. You come back and you go to your PI and you're a little down, and they say, "Come on, guy, this is normal. Go and look at those issues and come back with a stronger proposition or change it completely." And some of those propositions were eventually, panned out. I got grants out of those, and some of those ideas I've worked on since. So I think it's really trying to shape the kind of scientific questions, ask the right questions, set the right objectives, have the timelines and the scope. If you're going to do this and it's going to take you 5 years to even start, then it's not, you don't really want to, put effort on that. If that effort is going to be graded, because, as scientists, we judged, periodically. If you can't really deliver anything within a certain period, then you don't get the funding. So you have to really set your milestones carefully. And all those things, they try to teach us, but eventually, it's in the proof of the pudding. You really have to do the work, write the grant, and then get the comments and work on them. So I think those were some of the toughest in my early career, but I enjoyed them, looking back especially.
In hindsight, it wasn't. There are things you perhaps rose-tinted spectacles. We remember the good bits.
Yeah.
So Eric, what going through those early years, and even to today, were there any individuals in particular or groups that really inspired your work and the choices you made?
I think so. I mean, there was certainly, I mean, my lab at Hopkins definitely. So they just the structure of the research, the structure of the grants, and just the long-termism, if I could say that, of some of the ideas helped me. So effectively said, don't have short-sighted ideas. You can work on them, publish a paper, and that's it. But there's an opportunity cost to everything, and so do things that are going to define you in the long term.
And
Work on
who
you.
and who was your supervisor there?
So it was Zava Bujwala and Jerry Glickson, and I worked very closely with Zava, and I think that group obviously has carried on doing excellent work. I came off a little tangent from what they were doing. As you know, I started off my PhD doing a lot of magnetic resonance spectroscopy, and then shifted completely to PET, which at the time was the sort of PET against MR world. So I was one of those people who went very light, moving from the MR world to the PET world. Of course,
what inspired that switch? Because it's quite a it's
that
quite
switch.
great to make a leap.
It was a big switch. I think it was simple. It was because at the bottom of all that, I'm a pharmacologist, and it got to a point where I was struggling with sensitivity in vivo, that is, and I had made the decision that my career was going to be defined more by technological detection of what was happening in a living human being. And therefore, I had to develop those technologies, and I needed sensitivity. And PET offered the sensitivity, though not the resolution. And so the effort there was to bring in the understanding of how we could create the specificity alongside that sensitivity. And that encouraged me, at least I felt I could contribute to that field. And this, again, is important for any young person trying to get into any field, is how best their background can contribute to that field in a unique way. Because if you're able to define that, then that defines the trajectory of research that you do in a way, making you unique and therefore able to attract the grants and that sort of thing.
What was the biggest surprise of your career? I mean,
biggest surprise.
Did you expect PET to work as well as it did your work, or did anything you say wouldn't work has actually turned out to work?
So I mean, in my case, I started PET mainly from a sort of pharmacokinetics background, and so we would label drugs and figure out where they go, and there were lots of surprises. For instance, we found that tumors did not take up as much labeled drug as we thought they do. A lot of tumors just do not take up drug at all because of hypoxia and low flow to those tumors and that sort of thing. So, but having that evidence was important because it then catalyzed what we would do next to enhance delivery and to enhance specificity to tumors. I think the surprises were more about what happens when you translate from all the preclinical that we do into a clinical context, and then the stark realization that things are not the same. Some of our systems, I used to have discussions with Charles Keun about dead enzymes in tissue. So we'll spend a lot of time doing histology and various, and And especially with phosphoproteins, you get into an in vivo context and you realize that's not really what is happening as we've defined in a cell culture context. And I think it's a really careful translation of science from preclinical into the clinic. And some of those were surprises, but we went on to develop systems as you know, which have worked brilliantly. And I think the switch was more around moving a bit from pharmacokinetics and more into pharmacodynamics, looking at receptor expression, looking at very careful analysis of how to build up signal within a cell. So we then worked on not just on internalization processes, but also methods to trap various probes in cells through intracellular enzyme mechanisms. And we've built several of those now that have been translated from the basic understanding all the way into early clinical studies. We've got a lot of patents underpinning all these, and that has been, rewarding to us.
Is that what you've been the most proud of in your career? Is it that being able to get things from the bench into the clinic, or is there a particular standout moment or recognition that you're particularly proud of?
Yeah, I think that's, for me, that's been the highlights of my career. The idea of developing ideas in basic science and translating them into the clinic, and realizing that either things don't work or things don't fully work, and that the systems are very different. But it's actually quite interesting scientifically. And one day maybe I have to try and prepare a lecture on this of the translation from preclinical to clinical and the challenges that we face, because enzyme systems and receptors are not the same as we do the translation, and how we do the translation in order to not waste too much money, research grant money. It's in itself an art, I think. But yes, this is
so more art than strategy when it comes to choosing what to do and what to let go.
I don't think there's a panacea, but I think, as I mentioned, one element is niche. You have to define the niche. If your supervisor is doing the same, there's really no point. Find another idea. There are several. The other is to evolve. So be prepared to evolve. And that is more difficult. But I think even as a PI, our ideas have to evolve every 5 years. So we have to systematically change those, bring in new ideas. For me, it's been, what do I think in 10 years is going to be a major thing? So 10 years ago, I was thinking, a number of ways of detecting lesions in specific tumors that are, how to get to tumor will be important. Trying to roll out technology to the larger population would also be important. So we developed, for instance, the first fluorine-18 radio tracer to image somatostatin receptor. And that was not just by moving from one probe to another. We had to do a full screening and select new candidates. And so you have to have the long-term a little bit in mind. We're now working in brain tumors, which, a very difficult area. And again, the idea was to define new biology. And in this case, a new idea of the brain niche, what really stresses tumor cells when they get into the brain niche, whether they primary tumors or metastases. And a lot of this is also shaped by our evolving understanding of metabolism, which you are an expert on. And that is changing so much, not from just the genetic background, but the environment background. And so we're beginning to understand things that control the environment and make tumor cells able to undergo, make energy, undergo energetics and make nucleotides for DNA. So all of that sort of thing is shaping how we think, is shaping our proposition of what next to do effectively.
So your advice to evolve is, I think, a really important one, isn't it? And it's obviously part of the secret of your success. You've also been hugely successful at getting sort of more junior members of your group further in their careers as well. You've got advice for other PIs as to how to do that well?
Well, I think, certainly, and I remember when Jonathan came to give a talk, it's something that I think we drum into PIs here at Imperial, that, once you've sorted your career to an extent, you need to be supporting others. And for me, that is quite important. And, I'm glad that I think the basic thing has been, let's work to, in the same way, make sure that whatever we do, we're thinking like future PIs. And so these guys have left and obviously done extremely well. And I'm very proud of them. I think they've worked hard too. It's not just me. Yeah. They have worked hard, and some of them are incredibly hardworking. I'm not surprised that they got to where they've got to in directorship positions and professors. Some of them are professors already. So, I'm incredibly proud of them.
You mentioned Imperial's been working hard to have a positive culture and improve its culture.
Yeah.
Do you still feel there's significant discrimination, racism in academia? And if so, what do we do about it?
I think the emphasis probably is more on in equality, and this is a very challenging sort of turf to look at. But the unconscious bias or other can affect others coming up in their careers, and so I think that's where I think we should put a lot of the emphasis. How do we recruit and retain people across our sort of representativeness of the people in London, for instance, in UK in general, and so that sort of thing needs to be thought through and looked at. And I sit on a committee in the Faculty of Medicine that is looking specifically at this for BAME staff and that sort of thing. And I think it's important that we recognize that. I think it's important that the various managers in positions of authority recognize that as well. And so, the simple interview could have a bias if we're not careful. And I think the faculty is dealing with that, which is reassuring. But I think we need to, not sit on our laurels, and we need to work hard.
Yeah. So when you've got people coming through now at a more junior level, trying to make that transition from, say, particularly from the postdoc to that first fellowship or academic position, you've mentioned a few things. Think long term, be prepared to evolve. Be prepared to take on board criticism and react to it positively. Is there any other advice you would say to those folk to
I think maybe something that is specific to Imperial is walking the corridors and trying to pick up new collaborations. And it's I say that in very lightly, but it's important. There were times when you'll find me walking the corridors of South Kensington, talking to various people, arranging meetings with people in synthetic biology, and you know, I spent the day thinking, do I understand any of this? I had a collaborator who was working on the mathematics of boundaries. And eventually she became one of my collaborators on a CRUK center grant. And I didn't understand her maths, you know, to start with. But eventually I understood how those, you know, equations could be applied in the setting of cancer, and really to very useful endpoints. And it's that sort of trying to walk the corridors and pick up not just any collaborations, but long-term collaborations is very important. And I'm not talking about walking to a chemist and saying, you know, can you synthesize this compound for me? They are not interested. They are interested in developing some serious, you know, chemistry, not the one that you're thinking about. But if you go to them about a grant that you want to put together, of which they are part, and they can input into that grant, then that's a whole different ballgame. And I think that is important in Imperial, which is a sort of multidisciplinary environment. And if you like that sort of multidisciplinary science, then you should get off your backside and walk the corridors. And I think most people, you know, professors in Imperial do that in order to be successful. I think it's an opportunity for us. We see a little bit of that within the Convergence Center, but I think we have the opportunity to do even more as Imperial scientists.
Okay, well, that's, I mean, and your work is one of the best exemplars for multidisciplinarity and putting together different things. So I suggest if people want to get more inspiration in that direction, they can just look at your work and publications. Just to finish up, what's the single thing right now that excites you the most in what you're doing?
At the moment, I think right now one of the, I mean, there are several. Maybe I'll say two things. The first is the PET work that we're doing, and currently we're working on a compound called FPIA, which we're working mainly in brain tumors, and it picks up brain tumors and brain tumor metastases very well. And I think it has a future in various aspects of a disease that is quite tough. And perhaps we can not only make inroads into better detection and better characterization, both of which are very important in management of patients, but also develop a new area of theranostics, which are how to detect and treat. So you detect the ability of the probes to access and accumulate in tumors. And then if it does, you switch the radioisotope and you perform radionuclide therapy. So then it doesn't really matter where the tumor is, whether you can cut it out or not. It doesn't matter if you've got 20 lesions or not, you are able to target all of them by virtue of them harboring either a receptor or an enzyme that you can target. So that, for me, is quite exciting. I think I want to see the next maybe 5 years or so, or 10 years of my career in that arena. The other area that we've been working on, which I'm also really excited, is the fact that tumor architecture harbors so much more information than we can think of. Now, you know, one of the old NIH directors who sits on the Grand Challenge panel mentioned this and equated it to the London Transport Network, the tube. You know, if you take any tumor, regardless of what we teach our students, that tumors are just sort of some amorphous things, they have structure. And that structure is perhaps integrated not just from the genetics of it, but also the environment. And so that's how it's shaped. And we published some work, started with ovarian cancer, published some work detailing the fact that stromal contribution to the tumor environment shapes response, so that you can even detect who is going to live beyond 5 years or die within 2 years. And it's as stark as that, just from deciphering using artificial intelligence methods the tumor phenotype. And I think it's getting more exciting in the lab, and people are now even looking at more nuanced things relation to targets, specific receptors, just from looking at tumor architecture. I think this is the next big thing, and we're gonna see how one would be able to integrate this into prognosis. So prognosis, which used to be the preserve of genetics, is probably not going to be just the preserve of genetics any longer. And we can just take a CT scan that the patient has as part of staging and get much more information in terms of prognosis. Also in terms of prediction of whether they should have immunotherapy or not. I think we're going to get to the point where we can do this quite well with just basic substrates that occur just as part of the patient journey. So those are elements that I'm getting more and more excited about, and I've got a team of very smart people who are working in this area and trying to develop a huge selection of these in different tumor types. So those two excite me. They get me out of bed.
I mean, just hearing those two examples, I mean, everyone will get a sense of the diversity of work that you're doing. It is hugely exciting, and really look forward to seeing how that progresses. Thank you so much, Eric, for taking the time out to talk to me.
Thank you,
really,
Hector.
really fascinating, and I'm sure everyone else will enjoy listening to it too. Thank you.